The last couple sections that are chapter 3, we're dealing with sort of the nucleus. All right, so we've talked about the plasma membrane. We've talked about the cytoplasm, which contains the cytosol and the organelles, and now we're into the nucleus. Nucleus is basically part of the cell that directs the activities of the other cellular components. And it does that by determining types of proteins and how types of proteins that are made and how fast they're made, the production rate. Inside the nucleus, we have what we call DNA. You say DNA is what people can kind of know that's what makes you you, but what does DNA actually code for? It codes for the different genes in your DNA code for specific sections of RNA, which code for specific proteins, which help our body function in all types of ways. Remember, enzymes are proteins. A lot of things are made up of proteins. Proteins embedded in our plasma membrane. There's proteins in our mitochondria, so they basically help our body function. So DNA housed in nucleus contains code for nearly every protein in the body. Genes are within the DNA, so a gene basically is a section of DNA, are executed by several different types of RNA to build a wide variety of proteins. The nucleus consists of 3 main structures: the nuclear envelope. All right, so remember, this is a membrane-bound structure. We don't consider the nucleus an organelle per se. We consider it separate from the organelles, but it does have an envelope membrane that surrounds the nucleoplasm, which is like the cytosol of the nucleus. Water, free nucleotides, enzymes, proteins, DNA, RNA. We have DNA in the nucleus and associated proteins in the nucleoplasm. Our loose structural arrangement called chromatin, so that's DNA and associated proteins. DNA is not just a big long string. It's actually wrapped around certain proteins, so we call that chromatin. And one or more nucleoli, pair of nucleolus, site of ribosome biogenesis, suspended in the nucleoplasm. So we have this all going on in the nucleus. And we're going to talk about this, of course. All right, so the nucleus. Here you see the rough endoplasmic reticulum on the outside. That's an organelle. See the nucleolus, the nucleoplasm containing chromatin, DNA. Nuclear envelope, that's the nucleus, that's the membrane housing the nucleus. Nuclear pores, that's where RNA can exit the nucleus. And then we have ribosomes on the surface. Ribosomes are locations for protein synthesis. All right, so let's talk about the nuclear envelope. Double phospholipid bilayer, similar to that of mitochondria. Also, double phospholipid bilayer, that should ring a bell for the plasma membrane. Similar. Outer membrane, studded with ribosomes. Ribosomes, important for protein synthesis. It's continuous with endoplasmic reticulum, the ER. Inner membrane lines interior of nucleus, supported by a network of intermediate filaments. Intermediate filaments, remember those? Not the actin, not the microtubules, intermediate filaments. And then we have nuclear pores, large protein complexes, connect nucleoplasm with cytoplasm, allow substances to move between the two. One substance that's going to move between the two is going to be RNA. Very important. And this is just a diagram of the nuclear envelope. All right, so you have this double bilayer, right? Outer membrane, inner membrane. Double bilayer is important. We want to protect what is on the inside of the nucleus, which is our DNA. We don't want that mutating, obviously, hopefully. Hopefully not. Now, we have chromatin. This can get a little confusing. All right, so chromatin, one extremely long DNA molecule and associated proteins. This is how DNA normally exists in your body, unless something is happening, right? So your DNA, we know DNA is a long chain. We've already talked about DNA, but it's not just a long string like a piece of floss. DNA wraps around these proteins, these what we call histone proteins, and folds, it organizes and folds a molecule known as DNA to conserve space. Also helps prevent damage and reinforces DNA during cell division. So a nucleosome is a strand of DNA coiled around a group of histone proteins, right? So each one of these is is what we call a nucleosome. Right. So chromatin is a DNA molecule wrapped around its associated proteins. Each section is a nucleosome. Histone proteins, what they're called. During cell division, what we call mitosis, chromatin threads coil tightly and condense into chromosomes. That may sound familiar. Right? So chromatin, piece of DNA, is a strand of DNA with all of its histone proteins. During cell division, chromatin threads so it coils up real tightly and condenses into chromosomes. So chromatin and chromosomes are very similar. The difference is chromosomes are more condensed. When you condense all of our DNA into our chromosomes, human cells contain 2 sets of 23 chromosomes. One maternal, one paternal. You got one from your mama, you got one from your daddy. Total of 46. Sister chromatids, identical copies of each chromosome, made in preparation for cell division, connected to one another at the centromere. Remember the centromere when we talked about the microtubules? Right, so this is what it looks like. They can get a little confused when we talk about chromatin, chromosomes, histones, nucleosome, all right? But they're all related. So DNA, wound around its histone proteins. Each one of these is a nucleosome. Put this all together, you have chromatin. When it comes time for mitosis, it winds up really tightly and forms chromosome around its centromere. This is when it has been replicated and it's ready to be divided. So we replicate it, and then we divide it into a new cell, which we'll talk about that process. The nucleoli, the nuclear region responsible for synthesis of ribosomal RNA and assembly of ribosomes, right? Nucleoli, or the nucleolus. It's responsible for the ribosomal RNA, otherwise known as rRNA, and assembly of ribosomes. What are ribosomes? So remember, those are the organelles that help make where we have protein synthesis. Happens in our nucleolus or nucleoli. So what is this process of protein synthesis? I guess can get a little bit crazy, but we're going to try to go through it and you'll see this is something that happens very much involved in the nucleus, right? Remember, the plasma membrane is important because of transport into and out of cells. The organelles are important because they help the cell function, the functions of the cells. The nucleus is important because that's where we code for all the proteins. So protein synthesis, it's important to talk about that when we talk about the nucleus. So protein synthesis is the manufacturing of proteins from DNA. Like I said, we have gene expression. So a specific gene, a specific section of DNA that is coded into RNA, and then that's coded into protein. Right? So transcription, going from the gene, a specific section of DNA, to RNA. That RNA exits the nuclear pore, so exits the nucleus, binds to a ribosome, and then we have translation. Occurs in the cytosol, and that's an mRNA binds to the ribosome, and that RNA gets translated into protein. So you see the process here. DNA transcription, transcribed into RNA. This happens inside the nucleus, and then the RNA exits the nucleus, connects to a ribosome, and we have translation into a protein. This is very important, a very important process. You will see this on a quiz somewhere. Hint, hint. So a gene is a long chain of nucleotides. A section of DNA determines the sequence of amino acids in a specific protein. Right? So remember DNA has 4 different nucleotides, A, T, G, C. Each set of 3 nucleotides represents an amino acid. Okay? During transcription, each DNA triplet is transcribed into RNA. Each 3 nucleotide sequence of mRNA copy is a codon. During translation at the ribosome, so once we get out of the nucleus, each codon, so each 3 nucleotides of RNA are paired with a complementary tRNA, which is called anticodon. I know this is getting a little confusing, but each tRNA is bound to a specific amino acid. Remember, proteins are chains of amino acids. So remember, the protein is a chain of amino acids. The amino acid chain is coded by your mRNA, and each time a tRNA comes and matches the code, has the anticodon, matches the code of the mRNA, it brings with it an amino acid. And then the next 3 on the mRNA brings another tRNA, matches it, brings an amino acid, and those bind, and we have a chain of amino acids, and then that starts to fold, and we get our protein. So your genetic code is a list of which amino acid is specified by each DNA triplet. All right, so here's what it looks like, genetic code. All right, so remember in mRNA, in RNA, the thymine is replaced by the U. So we have the U in RNA, and the code codes for a specific amino acid. These are all amino acids. ALA, alanine. It's coded by a GCU triplet, GCC triplet, so on and so forth. We'll show this in a diagram. But mutations, changes in DNA, first of all, that can happen, but are due to mistakes in copy in DNA or induced by mutagens, which are agents that can change your DNA. ultraviolet light, radiation, certain viruses, cancer is based on mutated DNA. Right? So your genetic code is very important. Remember, it's in that double phospholipid bilayer in the nucleus. Your DNA is protected, but it can be mutated. The errors can happen, mutations can happen. All right, so let's go through this process in more detail. So transcription, going from DNA to mRNA, messenger RNA, we call it. All right. So we go from DNA to mRNA, which will eventually exit the nucleus through the nuclear pore. This process happens via RNA polymerase, which is an enzyme, speeds up the reaction, builds the transcript, binds to the gene, also brings in the complementary nucleotides, linking them together to form mRNA. Proceeds in 3 stages. Initiation. You're going to see the trend here, hopefully. So beginning of transcription, protein transcription factors bind to a promoter region near the gene. Remember, the gene is a section of DNA that codes for a specific protein. Right, so we have these transcription factors bind to this promoter region in the gene. RNA polymerase also binds to the promoter. DNA unwinds. Remember, RNA is single-stranded, so DNA has to unwind. We just copy one strand. DNA unwinds with the aid of the enzyme helicase. So unwinds the helix. Elongation, second step of transcription. RNA polymerase covalently bonds to complementary nucleotides to growing mRNA molecule. So it sort of opens up the DNA basically, and now starts forming a copy of mRNA. That's elongation by the RNA polymerase. Once we copy that section of DNA, copy that gene, you get to when the last triplet of the gene is reached, the newly formed pre-mRNA molecule is ready for modification. All right, so termination. We have that mRNA. Now this is called pre-mRNA because we still have to modify it a little bit. This is a video you can watch. Once we get the pre-mRNA, it isn't ready just yet. We have what we call exons and introns. So non-coding sections of the gene do not specify amino acid sequence. They're called introns. Sections that do specify amino acid sequence are exons. So we want to cut out the introns and just put the exons together. We have this process of RNA processing. Introns and pre-mRNA must be removed, exons spliced together. Now, when that happens, we can exit the nuclear pores and we're ready for translation, right? Then we have translation. It occurs at the ribosomes. Like I said, each codon, each 3 nucleotides on the mRNA codes with an anticodon, 3 nucleotides of the tRNA, and then that's connected, as you see here, to an amino acid. So we have the anticodon on one end of the tRNA. The anticodon is one end of the tRNA and amino acids on the other end. The anticodon codes with the codon on a triplet of the mRNA. All right, so we're just basically going down the code. So each ribosome has 3 binding sites for tRNA. So the mRNA goes to a ribosome in the cytosol. We have the A site, binds to the incoming tRNA. The P site The amino acid is removed from its tRNA, added to the growing polypeptide chain, which will eventually become the protein. And then we have the empty tRNA, then exits the ribosome from the E site. It's the exit site. And it's free to pick up another amino acid. All right, so it looks like this. Initiation, tRNA binds to mRNA. This is that mRNA that we got from the, that was coded for from the gene in the nucleus. Now it's in the cytosol at the ribosome where we have protein synthesis. It binds, brings a corresponding amino acid right here, the MET you see here. Initiation, elongation, next tRNA binds to open A site, allowing two amino acids to be linked together. First tRNA exits from E site, second tRNA moves to E site, now A site is open. Elongation, eventually you get to the end, stop codon, the last amino acid has bound, and now you have a chain of amino acids. Translation. And this is a diagram you can go over, and it puts it all together. And this is how we get from DNA to protein. Now, really quickly, the last couple slides, cell cycle. I'm not going to go through this in too much detail, but cells can't just come from nowhere. So we have what we call the cell cycle, order of events from the formation of a cell to its reproduction by cell division. So cell division is required for growth and development, right? Cells have to be able to divide so that we can grow, also for repair. And when cells get injured, old cells get removed, dead injured cells get removed, and we can build, body can have cell division so we can build new cells. I just wanted to have just a slide showing the phases of the cell cycle. Interphase, G1 phase, S phase, G2 phase, and mitosis fits right in here. See mitosis right in here we just talked about. And we're going to go through this in more detail in the future, but I just wanted to introduce it here. Your DNA is replicated here. It's important. Chromosomes, DNA replicated. Further growth, preparation for division. Your actual division, your mitosis is here. Cytokinesis, that's when you're splitting the cytoplasms, cytokinesis. And then we have cell growth here. The cell returns to interphase under certain conditions. Specific cells may enter a non-dividing G0 phase, but here. So this is the process cells are in. Your cells have to be able to divide, have to be able to produce more cells. That's how we grow. It's how we repair. Old cells get worn out, need to be replaced. So it's very important. Phases of the cell cycle.